Hello friends, this is your Yoganandam biology lecturer. In the video la நம்ம டிஸ்கஸ் பண்ண போற டாபிக் என்னன்னு பாத்தீங்கன்னா வந்து from biomolecules 11th standard syllabusல இருந்து நம்ம biomicromolecules already discuss பண்ணிருக்கோம், followed by biomacromolecules. So இந்த கிளாஸ்ல நம்ம டிஸ்கஸ் பண்ண போற டாபிக் என்னன்னு பாத்தீங்கன்னா வந்து biomacromolecules. So உங்களுக்கு எல்லாமே தெரியும் biomicromolecules அப்படினா ஆல்ரெடி என்னன்னு டிஸ்கஸ் பண்ணிருக்கோம். நம்ம biomicromolecules ஓட molecular weight வந்து less than 1000 Dalton இருக்கும் அப்படின்னு பார்த்திருக்கோம். இந்த molecular weight more than 1000 Dalton இருந்தா அது வந்து macromolecules அப்படின்னு சொல்றோம். So that is formed as retentate. Filter பண்ணும்போது cheese cloth யூஸ் பண்ணி ஃபில்டர் பண்ணும்போது நமக்கு அந்த retentate-ல இருக்கிறது வந்து macromolecules அப்படின்னு பார்த்திருக்கோம். சரி, இதுக்கு எக்ஸாம்பிள்ஸ் என்னென்ன கொடுத்துருக்காங்க பாருங்க. biomacromolecules இங்க சில எக்ஸாம்பிள்ஸ் எல்லாம் கொடுத்துருக்காங்க. என்னென்ன எக்ஸாம்பிள்ஸ் கொடுத்துருக்காங்க பாருங்க. Proteins, polysaccharides, nucleic acids are some common examples given for macromolecules. So the macromolecules oda molecular weight எவ்வளவு இருக்குன்னு பாருங்க. ஜெனரலா வந்து more than 1000 Dalton இருந்தாலே வந்து it is macromolecules அப்படின்னு சொல்லிருக்கோம். Proteins, polysaccharides, nucleic acids molecular weight வந்து 10,000 Dalton and above கொடுத்திருக்காங்க. So amino acids example for micromolecules பார்த்தோம். Many amino acids together form proteins. Proteins are examples for macromolecules. இதே போல glucose like sugar example for micromolecules அப்படின்னு பார்த்தோம். Many glucose molecule held together form cellulose, starch, that and all examples for polysaccharides. இப்போல nitrogen bases and nucleosides, nucleotides and all examples for micromolecules. Many nucleotides together form DNA and RNA. So DNA and RNA are examples for macromolecules. Let us discuss one by one. First, we are going to discuss about, say, the lipids. What do you know about lipids? Lipids வந்து retentate-ல form ஆகுது. Actually, lipids வந்து micromolecules சொல்றோம். ஆனா retentate எல்லாமே வந்து macromolecules தானே. Based on molecular weight வந்து lipids are classified under micromolecules that you try to understand. So lipids are not strictly macromolecules. So that means lipids are staying along with the retentate. Cheese cloth மேலேயே வந்து இது retentate-ல இருக்கு, acid insoluble pool-ல இருக்கு. But even though based on molecular weight, lipids are classified under micromolecules. So lipids are not coming under macromolecules. Then the average cell composition is given here. Try to remember. Normally cell contains 70 to 90% water, 10 to 15% protein, 3% carbohydrate, 2% lipids, 5 to 7% nucleic acids, only 1% ions. So from this part also question asked, try to remember cellular composition. Then let us discuss one by one macromolecules. So what is the first macromolecule is given? Try to remember. But proteins are given. What do you know about proteins? Whether proteins are macromolecules or micromolecules? Proteins are heteropolymers of amino acids. There are several amino acids held together form proteins. So proteins are examples for macromolecules. What is the meaning of heteropolymer? Made up of different types of amino acids. Proteins are made up of different types of amino acids. So it is called as heteropolymer of amino acids. How does amino acids held together? With the help of polypeptide. With the help of polypeptide chain. Polypeptide chains are linear chains of amino acids linked with the help of polypeptide bonds. How does peptide bond formation takes place? Formed when the COOH group of one amino acid reacts with the NH2 group of next amino acid. So where the two amino acids held together, the peptide bond formation takes place. See the diagrammatic representation. So here they're given is peptide bond in between two amino acids. In between two amino acids, peptide bond formation takes place. See the equation which is given very clearly. So here from one amino acid COOH group, another amino acid NH group, NH2. So from this one nitrogen from COAH, OH group held together by the removal of water molecule, peptide bond formation takes place. So due to dehydration, peptide bond formation takes place. Functions of proteins. Proteins carry out, there are several functional activities. Proteins are essential for growth and development of an organism. So it helps to repair tissues, act as carrier protein to transport nutrients across cell membrane, especially GLUT4 enables glucose transport into cell membrane contain transport proteins. So here one example is given, GLUT, GLUT4. So this protein helps to transport glucose into cell. Then some protein act as intercellular ground substance, example collagen. Then some proteins act as antibodies, fight against infectious agents. And சில proteins எல்லாம் பாத்தீங்கன்னா வந்து receptors act பண்ணுது to identify smell, taste, hormones act பண்ணுது, இல்ல receptors act பண்ணுது. Then some proteins are hormones, example insulin. Some proteins enzymes act பண்ணுது, example trypsin. Some proteins வந்து pigments act பண்ணுது, example hemoglobin. So இது எல்லாம் கொஞ்சம் clear understand பண்ணுங்க. Proteins வந்து carry out, there are several functions. So, especially proteins are essential for normal growth and development. So transport molecules and the functioning on the carry out the cell membrane la intercellular ground substance. Most abundant animal protein collagen. Try to remember. Then next paranga, most abundant protein enna just now nam sollirukom. Most abundant animal protein in collagen. Most abundant protein in the whole biosphere is rubisco, ribulose, bisphosphate, carboxylase, oxidase enzyme. It is commonly called as oxidoreductase enzyme. So this both the proteins also very important, collagen and rubisco. The next paranga, next on the structural levels of protein. Proteins are made up of, there are different levels. Some kind of proteins are made up of only one chain. Some kind of proteins are made up of, there are two chains like DNA. Some kind of proteins are made up of 3 chains. Some kind of proteins are made up of four different types of chains. So based on the number of chains, it is classified into primary structure, secondary structure, tertiary structure, and quaternary. Structurally, proteins differ from one another. Let us discuss one by one. It is the first one, primary structure. So what I said about primary structure, you try to remember. Primary structure have only one chain, only one polypeptide chain that held together with the help of peptide bond. So here amino acids are arranged in the form of sequence. The positional information in the protein. So this protein describes positional information in the protein. See the diagrammatic representation. Left end, first amino acid with the N terminal is left end, first amino acid with N terminal amino acids. Right end, last amino acid, that is C terminal amino acid. So here only one chain is given for primary structure. See the second one, secondary structure. So in the secondary structure, how it looks like, see the diagrammatic representation, it looks like in the form of double helical structure, like somewhat similar to DNA. The protein thread is folded in the form of helix. Only right-handed helix are seen in this structure. So this kind of protein structure seen in keratin, fibroin is always called as silk fiber அப்படின்னு சொல்ற நம்ம. So in the examples வந்து ரொம்ப important. The examples வந்து நீங்க recall பண்ணணும். Next, tertiary structure. The tertiary structure எப்படி இருக்குன்னு பாருங்க. Long protein chain இருக்கு. Also வந்து folded upon itself in the form of hollow woolen ball. இது என்ன ஃபார்ம்ல fold ஆயிருக்கு பாருங்க. hollow woolen ball மாதிரி இருக்குன்னு சொல்றாங்க. இது என்ன viewல இருக்கு? 3D viewல இருக்கு. It gives 3D view of protein. So in the tertiary structure வந்து பாத்தீங்கன்னா வந்து necessary for many biological activities of proteins. Example, myoglobin is important. So this kind of structure seen in which part of the protein? Myoglobin. So this one also asked in the examination. Try to remember. Tertiary structure looks like in the form of 3D view. The last fourth type is quaternary structure. Some proteins are an assembly of more than one polypeptide or subunits. Example, hemoglobin. Hemoglobin consists of, see the diagrammatic representation. Hemoglobin consists of how many units? 2 alpha units and 2 beta units. Totally there are 4 units. So here 4 subunits together form quaternary structure. So this type of protein structure seen in hemoglobin. So this also important one. Overall, the diagrammatic representation is given. Primary, secondary, tertiary structures. Let us go for next. Polysaccharides. First one, proteins are examples for macromolecules, followed by polysaccharides. What is about polysaccharides? Many sugar molecules together form polysaccharides. That means polymer of sugars, polymer of monosaccharides. For example, many glucose molecules together form starch. Many glucose molecules together form cellulose. Many glucose molecules together form glycogen. Many fructose molecules together form inulin. So starch, cellulose, glycogen, inulin and all examples for polysaccharides. Polysaccharides வந்து நிறைய sugar molecules இருக்கும். ஒரே ஒரு sugar இருந்தா that is monosaccharide னு பார்த்தோம். Many sugar molecules சேர்ந்து வந்து polysaccharides form பண்ணுது. இதுல சில complex polysaccharides கொடுத்திருக்காங்க பாருங்க. Complex polysaccharides வந்து made up of sugar with amino acids, that is commonly called as amino sugar. For example, N-acetyl glucosamine, galactosamine, glucosamine, these are all some common examples are given. So in the N-acetyl glucosamine, N-acetyl galactosamine, இதெல்லாம் எங்க இருக்கு அப்படின்னு பார்த்தீங்கன்னாக்கா வந்து so இந்த arthropods ஓட exoskeletonல இருக்கு. Chitin is the homopolymer of N-acetyl glucosamine. இது எங்க இருக்கு பாருங்க, N-acetyl glucosamine, exoskeleton of arthropods. Insects body cover ஒரு hard skeletal layer மாதிரி வந்து ஒன்று கவர் பண்ணியிருக்கோம். அதுக்கு பேர் என்ன சொல்றாங்கன்னா எக்ஸோஸ்கெலிட்டன் அப்படின்னு சொல்றாங்க. கிளியர் அண்டர்ஸ்டாண்ட் பண்ணிக்கங்க. எக்கினோடர்ம்ஸ் ஸ்டார்ஃபிஷ்ல எல்லாம் பார்த்தீங்கன்னாக்கா வந்து எண்டோஸ்கெலிட்டன் ஆசிக்கல்ஸ் இருக்கும். ஆர்த்ரோபோட்ஸ்ல எக்ஸோஸ்கெலிட்டன் இருக்கும். ஸோ இது ரெண்டுத்தையும் கன்ஃபியூஸ் பண்ணிக்க கூடாது, கிளியர் அண்டர்ஸ்டாண்ட் பண்ணிக்கணும். ஸோ நெக்ஸ்ட் பாருங்க, இந்த கிளைகோசிடிக் பாண்ட் எப்படி இருக்குன்னு பாருங்க. The glycosidic bond formed when the individual monosaccharides are linked between 2 carbons. So here with branch, without branch is given. The diagrammatic representation, see to that. The 2 carbon atoms held together by the removal of water molecule. Here also these sugar molecules held together by the removal of water molecules, the formation of glycosidic bond. So the diagrams you can clear understand பண்ணிக்கணும். How does glycosidic bond formation takes place? See the next, polysaccharides. Examples, starch, cellulose. Why starch with iodine turn into blue color, but cellulose is not giving blue color with iodine solution? What is the reason? Because of starch possesses several helical structures, say helical secondary structures. So with helical secondary structures, iodine hold and gives blue color. So இதுல நிறைய folded structures இருக்கு starchல, அதனால iodine starch ஓட bind ஆகி blue color கொடுக்குது அப்படின்னு சொல்றாங்க. But in cellulose there is no foldings, so there is no complex helixனா அந்த foldings சொல்லலாம். நம்ம foldings இல்லைன்றதுனால வந்து இங்க iodine cannot hold with starch cellulose, அதனால இது blue color சேஞ்ச் ஆகாது. ஸோ இதுவும் கிளியர் அண்டர்ஸ்டாண்ட் பண்ணிக்கணும். நெக்ஸ்ட் பாருங்க, nucleic acids, another macromolecule, nucleic acid. Nucleic acids are broadly categorized into 2 types. Try to remember DNA and RNA. DNA and RNA both are examples for nucleic acids made up of heteropolymer of nucleotides made up of, there are different types of nucleotides, adenine nucleotide, guanine, thymine, cytosine, like that. Many nucleotides held together form polynucleotide chain. இப்போ நியூக்ளியோடைடு அப்படின்னா என்ன பாருங்க, ஒரு பாஸ்பேட் குரூப், ஒரு சுகர் அண்ட் ஒரு நைட்ரஜன் பேஸ், அடினைன், குவானின், தைமின், சைட்டோசின், யூராசில் போல ஒரு நைட்ரஜன் பேஸ் டுகெதரா என்ன ஃபார்ம் பண்ணுது, நியூக்ளியோடைடு ஃபார்ம் பண்ணுது. பாஸ்பேட் குரூப், sugar and nitrogen base together form nucleotide. So a nucleotide without phosphate group is called as nucleoside. So rendu clear understand panninga what is nucleoside and nucleotide. Nucleic acids are two types. What are the two types of nucleic acids? Deoxyribonucleic acid and ribonucleic acids. Sorry, the secondary structure of DNA. DNA வந்து முதல்ல வந்து யார் வந்து இந்த டபுள் ஹெலிகல் ஸ்ட்ரக்சர் ப்ரொபோஸ் பண்ணாங்க அப்படின்னு பார்த்தீங்கன்னா வந்து வாட்சன் அண்ட் கிரீக் டிஸ்கவர் டிஎன்ஏ இஸ் பி ஃபார்ம் டிஎன்ஏ அப்படின்னு சொல்றாங்க. ஸோ வாட் இஸ் த மீனிங் ஆஃப் பி ஃபார்ம் டிஎன்ஏ? பி ஃபார்ம் டிஎன்ஏ ஒன் ஃபுல் டர்ன் வித் 10 ஸ்டெப்ஸ், 10 பேஸ் பேர்ஸ் இருக்கும். ஸோ அதுதான் பி ஃபார்ம் டிஎன்ஏ. ஏடிஎன்ஏ, பிடிஎன்ஏ, சிடிஎன்ஏன்னு சொல்லிட்டு டிஃப்ரெண்ட் ஃபார்ம்ஸ் ஆஃப் டிஎன்ஏ இருக்கு. பட் மோஸ்ட் அக்செப்டபிள் மாடல் அப்படின்னு பார்த்தீங்கன்னாக்கா வந்து வாட்சன் அண்ட் கிரீக்கோட பி ஃபார்ம் டிஎன்ஏ. சரி, டிஎன்ஏவில் எத்தனை பாலிநியூக்ளியோடைட் ஸ்டாண்ட் இருக்கு பாருங்க, 2 இருக்கு. ஒரு ஸ்டாண்ட் வந்து 5 to 3 டைரக்ஷன்ல ரன் ஆகுது. அனதர் ஸ்டாண்ட் வந்து பார்த்தீங்கன்னா ஆப்போசிட் 3 to 5 டைரக்ஷன்ல ரன் ஆகுது. ஸோ இந்த ரெண்டு ஸ்டாண்டுமே பார்த்தீங்கன்னா ஆப்போசிட்ல இருக்கிறதுனால என்ன சொல்றாங்க, ஆன்டி பேரலல் அப்படின்னு சொல்றாங்க. அரேஞ்ச் ஆன்டி பேரலலி ஆஸ் டபுள் ஹெலிக்ஸ். ஸோ இங்க பாருங்க, ஒன் ஃபுல் டர்ன் வித் திஸ் ஒன் டர்ன். One full turn with how many steps? 10 steps, 10 base pairs. Okay, so this is one nucleotide, this is another nucleotide. Two nucleotide from opposite stand held together form base pairs. So one turn with totally there are how many pairs? 10 pairs, 10 base pairs mean with 20 nucleotides. So what is the length of this one turn? 34 angstrom is given. Try to remember the data also important. So this is given in nanometer. In angstrom 34, in nanometer 3.4. At each step, see this is one step. At each step stand turns around how many degree? 36 degree. One full turn with 10 base pairs turn 360 degree. So that turning also try to remember. That in DNA, a nucleotide consists of nitrogen base, deoxyribose sugar, and a phosphate group. What is nucleoside and a nucleotide? Nucleotide consists of phosphate group, sugar, and nitrogen base. So phosphate group, sugar, and nitrogen base together form nucleotide. Nucleotide without phosphate group is called as nucleoside. So here the sugar and phosphate groups are forming as backbone of DNA strand. That also important. Sugar and phosphate group both are forming as backbone of DNA strand. These steps are formed of nitrogen base pairs. See this is nitrogen base pair. Nitrogen base pairs are forming steps. What are the different types of nitrogen bases which are found in DNA and RNA? Adenine, guanine, thymine, cytosine are present in DNA. Adenine, guanine, thymine, and cytosine. In RNA, thymine replaced by uracil. But in DNA, uracil is absent. In RNA, thymine replaced by uracil. See the diagrammatic representation. According to Chargaff base pair rule, adenine always pair with thymine, guanine always pair with cytosine, vice versa. So in between adenine and thymine, how many hydrogen bonds? Are present? 2 hydrogen bonds. In between guanine and cytosine, there are 3 hydrogen bonds are present. And phosphate molecule, how does phosphate molecule links the carbon atom, third carbon atom of one sugar and one nucleotide to the fifth carbon of the sugar of the succeeding nucleotide. See the diagrammatic representation. So this is phosphate group. Phosphate group linked to fifth carbon of one sugar. Then another side, when you are seeing the next, the phosphate group linked to third carbon. One side it is linked, phosphate group is linked with fifth carbon, another side it is linked with third carbon. See the opposite also. So first is phosphate group attached to third carbon. The next side is attached to fifth carbon. So that is clearly given with the help of diagrammatic representation. So here you try to understand between phosphate group and OH group of sugar. What is the name of the bond is present? Ester bond. So this is called as ester bond. This is phosphate group. Phosphate group, how does phosphate group is attached to sugar molecule, either side of the sugar molecule with the help of phosphodiester bond. Here two bonds are there. So one bond is linked to the first sugar. The next bond is linked to the second sugar. Like that, the succeeding sugar linked with one another. So that's why it is commonly called as phosphodiester bond. So here the ester bond is present on either side of phosphate group. So it is commonly called as phosphodiester bond. The bond between sugar and a nitrogen base is called as N-glycosidic bond. Next, so this one is phosphodiester bond. So what about this one? This one is N-glycosidic bond. Nitrogen base is attached to sugar with the help of N-glycosidic bond. So these two bonds are very important. Try to remember. So in the next class, let us discuss about metabolism. This is another important part from biomolecules. Thank you very much for listening our videos. Those who are watching our videos, you are requested to like, subscribe and share to your friends. All the best.